In this lecture, we will talk about the proteoglycan filaments as a spacer and role of proteoglycan in preventing rapid flow of fluid. Our this lecture is also in continuation with our previous lecture, the safety factors that normally prevent edema. And we are still in the factor number 1, which is basically the low compliance of interstitium in negative interstitial fluid range. Now we have discussed this point in detail many times, and we discussed again and again that the interstitium basically is in the form of interstitial gel. We also discussed this thing in detail that normally the interstitium is in gel, which means that outside the capillary and outside the cells there are proteoglycan filaments. These pink color fibers are basically the proteoglycan filaments, in which small amounts of fluid are present, but the fluid cannot move freely, especially when the interstitial fluid hydrostatic pressure is in the negative range. When this pressure, the interstitial fluid pressure, is in the negative range, when it turns into the positive zone, then there is rapid collection of fluid in the interstitium. This is something which we discussed in detail in the last few lectures. Now, the interstitium is in the gel form due to the proteoglycan filaments, and the gel form has its advantages in preventing edema and in preventing the fluid accumulation. But the interstitial proteoglycan filaments also has two other advantages in preventing edema. Now, one of the advantage of the proteoglycan filaments is that it acts as a spacer. Now, suppose for example, here is the normal interstitium. These are the cells, and these orange color fibers are the proteoglycan filaments in the interstitium. Now, this is basically normal interstitium, and this proteoglycan filaments basically acts as a spacer, or they provide some space between these different cells. If these proteoglycan filaments were not there, then cell would be like this. There would be no proteoglycan filaments and there would be no space. Now, the advantage of the space is that nutrients, ions and different nutrients from these distant cells, they can diffuse from the cells going to the interstitium, and through the interstitium they can go into the capillary. Similarly, nutrients from the capillary can diffuse into the interstitium, into these spaces, and the spaces are specifically provided due to or present due to the proteoglycan filaments. So the advantage is that nutrients and ions can move from the capillary lumen to the distant cells, which are basically present distant or at distance from the lumen of the capillary. Similarly, nutrients and ions from these cells can move into the capillary. Now, if these spacers, these proteoglycan filaments, if they were not present, then the nutrients and ions from these cells could not move into the lumen of the capillary, and nutrients and ions from the capillary could not move into these cells. Now, it would be very easy for nutrients and ions to move in and out of these cells, which are near to the wall of the capillary, but would be difficult to travel to these cells because the cell membrane will not allow easy movement. The cell membrane will never allow easy movement. But due to presence of proteoglycan filaments, there is movement of ions. Now, the proteoglycans, they will not allow rapid flow of fluid. They will not allow rapid flow of fluid, but they will allow diffusion. They will allow the ions and nutrients to diffuse, but they will not allow free movement of the fluid. So there is basically difference between free flow and diffusion. So nutrients can diffuse, but the fluid will not move freely. Now, the second advantage is that if there would be no proteoglycan filaments, then fluid would move. For example, we were discussing the diffusion of electrolytes and nutrients, but the free fluid is movement of the free fluid is basically prevented with the proteoglycans. If there would be no proteoglycan filaments as here, then what would happen is that if a person would stand up, now for example, we consider the cells in the legs of all the tissues in the legs of this person with proteoglycans. Suppose for example proteoglycans are present. If this person is standing, still the fluid present in the interstitium is loculated because the interstitium is in the gel form. And due to the proteoglycan filaments, the fluid cannot move freely. It is not allowing the free flow of the fluid. So the tissues remain intact. Now, suppose for example, if there would be no proteoglycan filaments and a person would stand up, then due to free flow of fluid, all the fluid in the tissues would come down and would cause edema, as in this picture. For example, there are no proteoglycan filaments. Here you can see the pink color is the cells, the orange color is basically the proteoglycan filaments, and the blue color is basically the fluid entrapped in the gel form of interstitium. But in this case, we have a hypothetical example, a picture in which the proteoglycans are not present, which normally is not the case. So what we see is that due to the absence of proteoglycan, there is free movement of fluid, and as soon as a person stands up, this fluid basically comes down to the feet, and edema is caused. Now, when there is increase in the interstitial fluid pressure, or suppose for example there is increase in the filtration, and there is collection of a lot of fluid in the interstitium, and the interstitial gel form is long gone, and there is collection of a lot of fluid in the interstitium, then edema occurs. And in such condition, when the patient raises their legs, then the edema can decrease and the fluid can come down. So this is because when the interstitial gel form is destroyed or is disrupted, then the rapid movement or the free movement of the fluid occurs, which basically leads to edema, which occurs in a lot of circumstances, as we have discussed the forces. The different forces. Suppose, for example, we have discussed these different forces. When the capillary hydrostatic pressure is increased, or plasma colloid osmotic pressure is decreased, or if there is a decrease in interstitial fluid pressure, all these factors can lead to a lot of fluid collection in the interstitium, which ultimately will lead to edema. And in such condition, if the patient raises their legs, then the fluid from the legs will come down because of their free movement, which occurs only in severe conditions. So, to summarize this lecture, the proteoglycans are helpful. The proteoglycans are helpful in making the interstitial gel, as well as they act as spacer for the diffusion, for allowing the diffusion of nutrients, and similarly they basically prevent the rapid flow of fluid and prevent edema in normal circumstances. So that's all about the first safety factor. Now here the first safety factor that normally prevent edema is finished, and in the next lecture we will discuss the second safety factor. Safety factor which prevent edema. So we have discussed in detail the first safety factor, which is the low compliance of the interstitium in the negative interstitial force pressure range. When the interstitial fluid pressure is in the negative zone, the compliance of the interstitium is very low, and in low compliance range it will not allow a lot of fluid to be collected in the interstitium. But the proteoglycans also play their role by making the interstitial gel and by preventing the rapid flow of movement in the normal circumstances. Thanks a lot for watching the video.